Sweet. So, we're recording now. I'm gonna go ahead and get started. I will be presenting my thesis on the dynamics of TVNS paired computerized cognitive training on working memory and visual spatial processing in healthy older adults. The principal investigator for this project is Dr. John Williamson, and my name is Xavier Velez, and I'll be submitting to the Department of Electrical and Computer Engineering at the University of Florida. All right, so let's go ahead and just kind of start off with a little bit of value and why this research is actually important. So first off, we'll start off with an observation, and that is that working memory, attention, and visuospatial processing decline due to aging. And that leads us to then an actual problem, which is that with these declines come declines in quality of life and independence that naturally decrease with age as a result of cognitive decline. Okay, and then pretty much I'm saying now that a solution is discovering an intervention that either prevents or reduces the effects of cognitive decline. And so more specifically in this work, we're going to be exploring the effects of TVNS paired with computerized cognitive training to see whether it has an effect on learning and memory. Okay, so how are we hypothesizing that TVNS actually impacts cognitive domains such as working memory or visuospatial processing? So TVNS is largely based on just invasive VNS. So the background for this comes from the vagus nerve afferent path through the nucleus of the solitary tract and the locus ceruleus, and those are primary regulators of norepinephrine. And so it goes to show then that that direct vagus nerve stimulation would be an upregulator of norepinephrine, which is also a regulator of neuroplasticity and long-term potentiation. And so the difference between that and what we're doing, though, is that we're doing a non-invasive approach to stimulation, where we're doing it through the posterior tragus, as you can see in this picture here, instead of directly sending a current to the vagus nerve. And so we're doing that as our primary intervention paired with computerized cognitive training. So this is just using a third-party service called BrainHQ that targets cognitive domains of interest, again, like working memory and attention, and it adapts to the user over time for dynamic training. So each participant undergoes 10 days of cognitive training with either TVNS or no TVNS. And we're trying to see if TVNS has an effect. So this kind of leads us to my main hypotheses. So the first one is that TVNS will modulate the functional connectivity in specific networks that are associated with the NBAC and UFOV exercises that are happening during task completion. So these are tasks that are happening within an fMRI scanner, and we're analyzing functional connectivity from while the participants are completing these tasks. And then hypothesis 2 is that performance on the NBAC and UFOV tasks can be predicted by right superior frontal gyrus volume for the NBAC and right superior parietal volume for the UFOV. And these are coming exclusively for pre-intervention. So this is without TVNS as an effect. Can we say, all right, you're going to do better or worse on task performance based on white matter volume in these regions? And then the third hypothesis is kind of bringing it all together to say that TVNS induced functional connectivity changes that may be identified in hypothesis 1 will be reflected in NBAC and UFOV performance, regardless of the effects of white matter volume. So if someone has decreased superior frontal gyrus volume that is found to affect their performance, I hypothesized that regardless of the change in white matter volume between participants, there will be a positive effect on performance as a result of
So,
CVNs.
so I'm gonna interrupt you and ask you a question.
For sure.
So what is white matter?
Yeah, so white matter, those are the I guess like primary like main regions like in the brain. So those are what's doing I guess kind of like the heavy lifting in the back. And that's what we're analyzing.
Okay.
Yeah.
Yeah. So uh, white matter is can be thought of more as the wires that connect areas of the brain together that do the heavy lifting processing.
Oh, I see,
So
I see.
these are the pathways. So actually, what you've got, and it's probably this will need to be edited in your manuscript. So
I see.
the regions that you've picked are actually gray matter regions.
Okay,
So
I see.
the white matter is the interconnections. So for example, if you're interested in um, you picked the right region. So
Hmm,
parietal lobe and dorsolateral prefrontal cortex are primary hubs for executive functions, right? So what you're interested in is the volume of changes in those regions associated with aging. That's what you've looked at in your analytic model, right? You could also look at connectivity from white matter, which would make a lot of sense, but you would be looking at a different structure called the superior longitudinal fasciculus, which connects dorsolateral prefrontal cortex to the parietal lobe. They're different research questions.
I see. Okay. All right. Good to know. Thank you. Thank you. All right.
You're welcome.
Awesome. All right. So kind of moving on, and I'll try to keep that in mind when talking about
Yeah,
this
you have to
because
why
yeah.
here.
Yeah. I
Yeah. Yeah.
all right. So kind of breaking down each of these hypotheses now. Just a reminder, the first hypothesis is that functional connectivity will change as a result of TBNS in both NBAC and UFOB exercises. Um, so for this hypothesis, it might be important to first start by saying that it's typically increased functional connectivity that is linked with higher cognitive ability. And that's going to be a theme that we kind of challenged throughout this defense. But a similar study exploring the effects of transcranial direct stimulation or TDCS actually found changes in functional connectivity as a result of stimulation. So there is I guess, precedent of.
Meaning they found after stimulation over time decreases in functional connectivity.
So actually, after taking another look, they ended up actually finding increases in functional connectivity. And I know we briefly talked about that it
Yeah,
looked
the
at a
difference between
glance.
metabolic rate and functional connectivity. That's right.
Mhm, mhm, yeah. Um, so so yes, but there might be some I guess interesting things in play that again we'll kind of discuss throughout the defense. And then it is also maybe important to note that the regions that we're going to be analyzing are going to be specific to NBEC and UFOB. So these are going to be regions that might be um, most relevant to either working memory for NBAC or visuospatial processing for UFOB, and that intersect with norepinephrine projections. Okay. Now, for the second hypothesis, this is going to be that performance on the NBAC and UFOB can be predicted by these, I guess, gray matter volumes. And so I guess in your mind, maybe just replace white with gray from here on out. But working memory is linked to, I will just say, the integrity of brain regions in the prefrontal cortex and the integrity of regions in the parietal lobe for visuospatial processing. And there are also significant negative correlations between age and integrity of volumes. So volumes decrease with age, cognitive domains decrease with integrity, right? Or as integrity decreases. So we can essentially just say aging affects the cognitive domains that we're interested in. So kind of just breaking down these two a little bit further, the right superior frontal gyrus is linked to working memory and attention. So again, that one's going to be for the NBAC, and it does partially overlap with the dorsolateral prefrontal cortex, which again, as Dr. Williamson mentioned, is another one of those key areas. So it's kind of a good one to pick. And another thing that's actually really cool about this one is that it's prone to age-related white matter atrophy. I guess gray matter atrophy would be more relevant to say, possibly. And then for the UFOV, we have the right superior parietal lobule, and that is linked to visual-spatial processing, again, UFOV. And this greatly atrophies in people with dementia, which might not be super relevant because, again, this is people who are healthy, but maybe still interesting to note. Okay.
Going back to the third research question, I just want to make sure I'm on track. When
Mhm.
you say integrity, you're talking about the output of your functional MRI?
No, so this is purely volume, actually. So this is
Yeah. So
whether,
it's also a term problem. So you need to consider Xavier, the difference between uh, basically neurodegeneration or loss of gray matter volume and changes in the integrity of a network.
okay.
So white matter implies network, which is where the confusion is here relative to this integrity concept. What you're saying is that uh, differences in the behavior of prefrontal and parietal networks is associated with reductions in working memory performance. That could be caused by either decline in or decrement in volume in prefrontal cortex or decrement in volume in parietal cortex associated with dysfunction in those brain areas, or with connectivity between them affecting function. So that's the functional connectivity issue. So in the fMRI test, you can talk about network integrity. In these gray matter analyses, it's more about changes in the underlying brain health of those regions.
That makes sense. Okay. All right. Awesome. Awesome. All right.
So just out of curiosity, how do you guys measure volume? Is that
Uh, yeah. So there's a um, program or set of algorithms called Free Surfer that essentially automates the segmentation process, and then we just correct for total intracranial volume at the end. So it's essentially a percent of whatever region we're looking at, compared to how big their head is. So
Yeah, so it's MRI. You can
yeah, yeah.
use Free Surfer is a one that adapts to the subject. So it's an individualized, as you say, intracranial volume or cortical map. There's an atlas to identify the specific gray matter structures of interest, and that is then applied via this automated programs, Free Surfer, to get a quantification of that.
All right. So, kind of moving on from there, just further breaking down that third and final hypothesis, where we kind of bring them all together. We are saying that, or I am saying that CVNS induced functional connectivity changes that are coming from the first hypothesis will be reflected in NBAC and UFOV performance, regardless of potential neurodegeneration of gray matter volume. So kind of just process here. We're saying that CVNS modulates functional connectivity, and then the potential age-related effects of volume degeneration are negated by TVNS, and then those changes are reflected in task performance. So these are my three hypotheses. All right. Just going to do a brief data description just to give a better understanding of what the data we're working with is. So the key points here are that there is an MRI that all participants undergo right at the beginning, and that is where our pre-intervention assessment is coming from. So throughout the defense and in the thesis, you'll hear the terms like pre-intervention, post-intervention. So this pre-intervention is before they even get randomized into groups. They undergo an MRI and they perform both the UFOV and NBAC in the scanner. So then they're randomized into the TVNS and sham group. They undergo 10 sessions or 2 weeks of cognitive training, either with or without TVNS. And then immediately after, they undergo their post-intervention MRI, where they again do the UFOV and NBAC tasks. And again, those are going to be our post data. Okay, and then kind of just a brief, again, kind of description of the data, just to give you a good understanding of what we're looking at here. So we ended up with total sample size, at least for this defense, of 23 participants, which is a pretty limiting factor throughout. So I guess kind of just keep that in mind throughout the defense. But we didn't see any significant differences between groups for the age, education, or these standard just like cognitive tasks that were done during screening. So all this is to say the groups were even at the start of cognitive training. There weren't any statistically significant differences between the groups. Okay, so now I'm just going to briefly describe the tasks and just how these, are completed within the scanner. So the UFOV, again, this is the one that has to do with the visuospatial processing. It has 4 major stages. First, it gives you a cross in the middle of the screen, and then either a car or truck is presented in the middle of the screen, while an additional car is shown in one of the 8 octants surrounding the middle. And then there's just like a mask that just covers everything. And then finally, there is the probe stage where either a car or truck is shown in the middle, and then a car is shown on the peripheral, again, in one of those 8 octants, and the participant has to press the button in the scanner if the vehicle in the center is the correct one that was shown in the stimulus, and the car in the periphery is in the same position as it was in the stimulus. And they do not press the button if they're different. So that's the UFOL task, and then the 2-back. And so I'm just gonna go over the 2-back instead of the 0-back because that's the one that I'm primarily interested in. Just a little note here. The 0-back is mainly used as a metric of processing speed rather than actual working memory.
It's also a contrast. It's not very difficult.
Right.
What was the
Yeah.
last number I said, right?
Yeah, exactly. So
The
analyzing it.
the UFOB test is testing what? Like, some of these tests, like they test declarative memory, associative memory. What does the UFOB, if you had to put it into words, it seems like a really cool test. What is it actually evaluating?
Yeah. So yeah, that's I guess the task that I'm referring to when I say visuospatial processing. So it's pretty much whether they can be focused at one point in the center and be able to identify like objects in their peripheral vision correctly.
Yeah, de acronyming that probably is helpful, right? Because
Yeah, for
it's
sure.
useful field of view, right?
Right. Yes. Yeah.
Mhm.
So yes, that is the useful field of view task. And then the 2-back is fairly straightforward. So this one is more in line with working memory and attention. And there is an alphabetical letter that is presented. And then you have a cross in the middle of the screen, and a new letter is presented after that. So the main goal is just to be able to identify whether the current letter you're looking at is the same as the letter that was 2 letters back. So, it's pretty well depicted in this image here, where the L would be a correct target. And in this case, after seeing the L, you would press the button in the scanner, whereas for the S, E, and B, you would not press the button in the scanner. Okay, so gonna kind of move on from there and kind of just go into some of the methods that I actually use to evaluate all of these hypotheses. So for the functional connectivity analysis, first we took the raw DICOMs from the scanner, put it into BIDS format, and then pre-processed it using fMRI prep and smoothing and con. And this just gets rid of things like movement, heartbeat, hopefully, and just anything that might disrupt the quality of the scans. Then we did a little bit of denoising in con. And then in this stage, this GPPI maps, that stands for Generalized Psychophysiological Interaction Maps. And so essentially what that's doing is it is taking every single part of each task. So for example, in the NBAC here, we have the 0 back pre, 2 back pre, 0 back post, and 2 back post. So for each of those stages or sessions, we'll call them, it runs a bivariate regression and then maps the functional connectivity as a separate matrix for each of those conditions. And then this final step is at the group level. It takes all of those matrices and then evaluates the differences between the groups. And when I say differences between the groups, for the UFOV task, I am comparing the host, wait, here we go. So the host probe to all of the other stages. And then in the NBAC, I am comparing the host 2 back to all of the other stages. And then also just another note, both groups are corrected for age, sex, and education within the GLM that's at the group level. Okay, so kind of going into the actual results for functional connectivity. This is where most of the exciting results are coming from here. So first off, we saw that in the right supplementary motor area, and this is an ROI to ROI analysis within con. So that's essentially saying that this region of interest had enough change in connectivity within the rest of the network to have like a significant effect that we can report. And so we see between the right inferior temporal gyrus and left inferior temporal gyrus, there is a significant decrease in functional connectivity. And again, I mentioned at the beginning that typically functional connectivity, you'd expect an increase in functional connectivity if you want to see an increase in performance.
Are you so I've been popping articles at you. I don't think it's quite that definitive.
Right.
So you'll want to maybe look for some meta-analyses relative to this, because there's some data with aging where decreased functional connectivity is associated with better performance.
Right.
Yeah. So I would still see this as a developing area, and one must consider which networks and the context, right?
Absolutely. Yeah. You're kind of spoiling my my end my
Oh,
end
I'm sorry.
aha
It
opinion,
was
but
it
no,
was the
I'm
use
kidding.
of
I'm
it
kidding.
was the use of typically that
Right.
sparked me. My
Sorry about that.
apology.
Yeah, that was probably not the best way to go about that. So but yes, no, that is exactly correct. So even though on first glance maybe these might spark some alarms from a younger population, especially in an older population, we'll see later that there are some really interesting things that this might allude to. And then in the two-back functional connectivity, we actually saw a whole bunch of decreases in functional connectivity, which again, really great. It also might be important to note that some of these in here do have FDR corrected p-values that are above 0.05 but below 0.1. And because of the lack of data that we have with our 23 participants, we decided it might be best to just give a little wiggle room, for some maybe weak correlations right now to be possibly further, like, I guess, bolstered by more data in the future. In this two-back functional connectivity analysis, it is also important to note that while the UFOV was an ROI to ROI analysis, this two-back is actually a cluster level analysis. So instead of saying, hey, this specific region, this right supplementary motor area is an important region of interest, instead it's saying, all right, all of these regions that are identified in the seed column, these are within a cluster that the tool that I use has identified as like related regions. And I'm decently certain that it just partitioned those by left and right side. So it's saying essentially on the right side, there were a lot of changes in connectivity to other parts, both within the right and the left side.
So I have a question on
Yeah.
the tool and how this works. So you have a region of interest,
Yes.
and how many voxels are there? And how do you map a particular region and say, okay, this is this region, this is this region, I'm looking at this connectivity. How does that determine statistically? Because obviously, I guess you're doing some type of statistical t-test, or what are we looking at here with regards to the p-value? Because, I think we talked about this in my class, where, if you just keep running t-tests,
yeah,
eventually something's going to come statistically relevant. And all I'm seeing is like one little point, and I doubt that's the case. So I'm just trying to, could you explain a little bit more towards that and how this is statistically relevant?
Yeah. So I'll kind of start off with kind of just the first section of like, the maybe number of voxels, or like how we're actually breaking down the regions within the tool that we're using. There's like a standard for I guess like the placement of regions, and I guess it's called kind of like MNI space. At least I think I'm referring to it correctly. And so within the tool that we use, we have just XYZ coordinates, and then I'm using spherical ROIs. So I give it an XYZ coordinate and then a radius of voxels. And so for most of these voxels, at the end I have some actually.
So the number of
slides.
voxels depends on what?
So the number of voxels, well, it depends on how large the region is expected to be.
Also the resolution of the MRI, right?
Yes, yeah, and that as well.
It's one of the problems with the higher strength MRI. So like some of the 7T ones, you're vastly increasing your
right
comparison.
I see. I didn't know there was that big of a difference in some of these. That's good to know. Well, I guess you'll be happy to know then that these ROIs are from the exact same scanner that we use for this study. So this is just an example of, we have our XYZ coordinates and then a radius. And actually, sorry, I may have misspoke. Radius is not in voxels, it's in millimeters. And so I guess then that is transformed into voxels like within the tool. Okay, and then back to how we're actually evaluating this.
So the region of interest is a set of voxels, and then you're looking at the average value that's produced there, intensity tied to that.
Yes, yes, that's correct. That's correct.
Okay.
And for
And so
connectivity specifically, why is it connectivity versus, say, activation in a specific region?
yeah, yeah. So that's where kind of these two models at the end kind of come in. So again, like the tool that I'm using, Con, it's not necessarily looking at like how do I say this? Like I hate to use the term to describe the term, but it's not necessarily looking at activation. It's instead looking at kind of like the synchronization between the two regions during the task completion. So then when it's doing this regression, this initial regression, it's taking each region of interest and it is fitting it to a a general linear model against every single other region of interest within the network. And so that's the first step. And then in the second step, it compiles all of those. So for each participant, it'll have 4 different matrices, and then it combines each of those matrices that correspond to the same section of the task. That's so wordy. So I can break this down further if that didn't make sense. But it combines all the same sections of the tasks within participants and then runs another general linear model. So when we get the p-values here, these aren't necessarily like t-tests that we're running.
Okay.
It's yeah, yeah, it's the fit of the model and the effect size of um uh this end GLM.
And you're controlling for the multiple comparisons in 2 ways, right?
Yes, exactly, exactly. Yeah. So I'm controlling for pre and post, and then between the 2 different areas in each task. So for end back, that's
Yeah,
comparing between.
using voxel and cluster
Yes, yes, exactly, exactly.
FDR,
Yes.
right?
Yeah,
Yeah.
that's correct. And so in the UFOP, there were no significant results that came out of cluster level, but there were the significant effect that came out of ROI level
So
in
I'm
the optics
confused by
GLM.
that. I noticed that in your draft.
Mhm.
Um, so these p-values incorporate both the voxel and the cluster threshold, right?
No, no. So
No.
so the
Okay.
UFOB is only reporting the voxel level, and
How did
then
you how did you do that? Because don't you at least in my experience, you specify both levels.
in the analysis page, like where I got all of this data, there you can toggle it. So there's like a little drop down menu, and you can literally just select either cluster level or ROI level. Um,
Isn't it
and so,
both? You
um,
set one at 0.05 and one at 0.1 or
for the thresholds, yes, but for the actual test that's
oh,
being.
you're talking about, I'm sorry, we're thinking about two different things.
Oh, I
Thank
see
you.
what you were saying.
Yes.
Okay. Okay. Yes. So for the threshold, yes, it is 0.05 for the to determine. Okay, let me make sure I word this right. For the ROI level, it's using a 0.05 FDR corrected p-value to determine whether the overall region or cluster of voxels has significant changes in connectivity. And then a corrected p-value of 0.1 to determine whether the individual connections within that region are significant. And then on the cluster level analyses, it's using a 0.05 value, again, 0.05, to determine whether, like in this case, the right side of the brain has a significant amount of um, connections that are coming out of it, or a significant strength of connections that are coming out of it. And then a 0.1 value, which is kind of reflected here, where it is determining the individual connections between one region to another. Again, that was super wordy, but I hope that made a little bit more sense. Um, okay. All right. So yeah, I'll move on to the next hypothesis if you guys are both okay with this part. All right. Cool. So, man, I'm really wish I did. I really wishing I didn't say white matter so many times within this. It's like pretty much on every slide from here on out. But
I could have allowed you to blissfully continue through without interrupting with that. Yeah.
no, I'm glad you did. I think it's important for me to know and I guess consciously correct throughout this. So, okay, so how we actually determined the white matter impact on the performance. So first, took the raw DICOM, put it into bids. That's the same as the last hypothesis. And then again, this is what I was talking about where it's free surfer. So there are set atlases that are used to take each individual participant, take their structural scans, and then segment their like regions into the different volumes of importance. And then again, as I was saying before, we correct it for intracranial volume. And then here is where I actually validate that the regions I selected before are actually meaningful, that they're the right regions to test. And I will actually say at this point that I selected these regions like way beforehand. And I'm pretty happy that it ended up actually validating that they're
Yeah.
correct.
And you selected them how?
Uh, whole lot of research and
Yeah.
literature reading. So yeah, it was just a lot of papers. And yeah. Okay, and then at the end here, we're going to take the regions that are identified as important from this regularization, and then we're going to see whether white matter, or no, whether the gray matter has, the gray matter volume can be used to predict performance. So again, kind of just a note on this, all of this is using pre-intervention data. This is not using TVNS.
So can you talk a little bit about Lasso?
Yeah, absolutely. So Lasso
I guess
is,
we're doing questions while you go. It just seems we kind of organically.
yeah, that's perfect. That's perfect. Yeah. So Lasso is again regularization. So in a standard like linear regression model, like I'm using here, the objective is to minimize the error, right? So what Lasso does is it adds at a term to that objective function, and it says, hey, let's add onto this function that we're trying to minimize all of the weights that we've applied to this fit model. And then because it's a minimization function, the model is then prone to giving the smallest weights that it can. And that's regularization as a whole. But then lasso in particular is used oftentimes as a feature selection method. So it's actually, it's defined as like lambda times the sum of the absolute value of all the weights. And the proof for that is a mess. I gotta be honest, I might not be able to recite it for you. But essentially that absolute value term makes it so that it's more prone to just cutting features out rather than just minimizing them to just a really small value. So especially as lambda increases, which is the term that's saying essentially how harsh we want the lasso regularization to be, it'll start cutting more and more features out. Okay,
So are you familiar with other types of regularization or?
I've used ridge regularization as well, which is instead of the absolute value, it's the square of the weights. But other than that, I haven't used other regularization methods.
So if you're trying to predict, why not use ridge?
Well, because ridge, that's maybe an important distinction. So this step of regularization here, I'm not necessarily using that in the final model. I'm purely using this lasso
Just for the
regularization
feature
as a feature
analysis.
selection. Yeah.
Okay, great.
Ridge doesn't.
And lasso is so the reason that lasso came to my awareness was that we kind of liked in these big cognitive type questions where we had multimodal imaging data and lots of different cognitive tests and old people and medication issues and all a high density variable space, basically, these selection models. And we had previously been using stepwise regression, but statisticians don't like it very much, right? So I was looking for things that would allow us to do selection model type things. What's important in this relative to this cluster of variables, and lasso was something that was recommended by a statistician here, and a few things relative to that, as Xavier pointed out, the regularization model were approach were pointed out is useful there. The one that the statistician used, and to me this sounds like magic, is the oracle property for the adaptive lasso approaches that they do, which I still don't understand that, but I'm a psychologist, so what do I know?
Yeah. No, so usually lasso is great for determining and interpreting which features are important.
Yep.
And we use that, so our lab is very signal processing based. So we're always trying to evaluate, okay, we developed this new signal processing tool. We believe it provides more information than these classical techniques. How do we prove this, right? And so we use lasso and ridge and other statistics to evaluate what, is there more information gain in describing a phenomenon. So I just wanted to see how, I was just
I see. testing our student.
Yes. All right, awesome. Sweet. And then boom. Yeah. So just a little more on the lasso. So the 8 ROIs that were tested were the left and right hemisphere, side of the superior frontal gyrus, superior parietal lobule, and then frontal pole and temporal pole. Okay. So again, we're going to start with the UFOV results on this. And again, here you can see the lambda. And so again, this is not that lambda is equal to 90, but the function that I use uses just 100 lambda values and calculates the weights for all of those values. So on the 99th value, which is going to be the highest in that model, it ends up fitting it to only one region in this case, which is great because that means I only have to put one region in the model, and that's good. And it just so happens that that region that I identified is the right superior parietal lobule, which is the one that
isn't that coincidental?
I know, right? I was actually I was very, very happy about that. So yeah. And then it kind of went downhill from here, just in the sense that my hypotheses were not found to be correct. So using a GLM to then try to predict performance purely based off of right superior parietal lobule volume, correcting for age, it did not have any significant effect. So you can't necessarily just look at that volume and say, hey, you're going to do great on this task or not great on this task. And
so what is the correlation between age and right superior parietal volume?
Yeah, so it does decrease. Volume does decrease with age. That being said, though, it typically decreases with age more so in unhealthy older adults, so people with dementia. Whereas the right superior frontal gyrus typically decreases in volume a little more, even in healthy populations, than you would see in the right superior parietal lobule. So that was one of the biggest things that I thought was really interesting about the superior frontal gyrus, just that even in healthy older adults, there was a difference in volume seen in literature. Okay, so did the same thing, and again, came out that the region that I identified for the 2-back was on top, which I love to see. And then, so for this one was actually interesting. I initially ran it and I ended up with like really great results. I had a p-value of 0.002. I was, super excited. And then I plotted the data just because that's a pretty big result. I wanted to make sure that I wasn't, skimming over anything important. And there was a possible outlier. There was a single participant who had a very high two-back score, but a very relatively small superior frontal gyrus volume. And so I removed that singular participant, reran it, and then it wasn't significant anymore. So that being said, 0.163, that's a little closer. So I would be actually really interested to see whether this one holds up. I think especially because of the aspect of this region degenerating at a more regular rate. But as far as the participants here go, I think it's safe to say that removing the outlier was the correct, or then provides the correct fit. And so, yeah, that's that one done and dusted. And I was able to conclude that the gray matter volumes do not impact or cannot be used to predict task performance. Okay, so oh my animations are a little messed up here, so I'm just gonna go ahead and put them all out there. Okay, so on the last one where I'm kind of bringing it all together, there's kind of two aspects of this that I wanted to test. So this first one at the top, essentially this is just testing if we correct for age, sex, education, and TOPF score, which is, again, it's just the test of pre-morbid functioning. It's just like a word test, standard cognitive performance task. So really, this top one is just saying, does TVNS impact performance? And actually, something really interesting that I ended up doing here is that because there are only 2 occasions, I did not use a change score as the dependent variable. And instead, I performed just like a simple t-test on the pre-intervention performances between the groups. And if those weren't statistically different, then I didn't include the pre-intervention performance as a covariate in the final GLM model. So that's that one. Wait, sorry, were you about to say something?
So given the small sample size, wouldn't you want to take advantage of the increased power by having the 2 cognitive time points?
Yes, I suppose so. One thing that I did end up seeing though is that when I used wait, sorry, okay, I guess let me would you more so say that you're referring to taking the change score as the dependent variable or adding the pre-score as a covariate in the model?
Well, you could do it as a repeated measures type approach, or a change score, either one. I mean,
Okay.
mathematically, it'd probably work out pretty similar.
Yeah, I will say one thing that because I tested this with everything just to see what was going on. And I did find that when you added the pre-intervention score, it fit the model really well, but with an effect size that was almost exclusively attributed to the pre-intervention effect. So the other variables weren't necessarily being tested. It was just saying, okay, here's your pre-intervention. And then here's the post from that. And I think that's primarily because in general.
So basically the bottom line was that the pre-post performance on the UFOB was not very different. Is that right?
Yeah, exactly. That's correct. That's correct.
Mm-hmm.
Yeah.
Yeah.
So yeah. So again, this first one, just calculating whether TVNS impacts performance. And then in this second one, here's where I incorporated functional connectivity into the mix. And I also just controlled for the gray matter volume, but that didn't make much of a difference anyway. So in this, I used a kind of thresholding algorithm. So those matrices that I talked about before, where they had the 10 by 10 matrix of all of the weights fitting each ROI to each other ROI. I essentially exported those and then calculated for the stage of interest. So for the NBAC, it was the post-intervention 2-back. So I essentially said, all right, so for the post-intervention 2-back, how many regions within each participant had a effect or a weight that was bigger than 0.5 for the UFOB and 1.6 for the 2-back when compared to the other 3 stages. And so that was kind of just my way of extracting the feature from the functional connectivity weights. So it might be important to note that although this does take the same models that were used in this first hypothesis, it is not necessarily taking these exact values from these functional connectivity analysis results and importing those into the GLM.
So does reduction in functional connectivity predict better performance or no?
So it does not. So it sorry, hold on. Let me make sure I'm saying this right. Increased functional connectivity was correlated with increased performance, but with maybe some odd statistical caveats. So kind of breaking that down, in the interaction between stimulation group and functional connectivity alone did not have a significant effect on performance, which kind of makes sense, and that'll kind of support a little bit what I'm referring to later. And that is also somewhat reflected in this graph here, where let me kind of start from the top here. So first off, TVNS alone was not found to have to be a predictor of task performance. So just based on whatever group they were in, that wasn't enough to say, hey, they're going to do better or worse, just purely based on performance, right? And then that's when I added the interaction term between functional connectivity and the stimulation group. And there were significant results. Excuse me. There were significant results here, and we have a p-value of 0.006. But after breaking up each group and running a separate GLM for both the stimulation group and the sham group, I found that functional connectivity was the main predictor for both models. So essentially what I'm saying here is that there was a little bit of a disconnect where there was something in the functional connectivity analyses that was allowing us to predict performance, but not when it was mixed with TVNS. Or sorry, not when it was evaluated with TVNS. So yeah, so that was UFOB. And then 2-back, essentially all of these were not significant. So again, there was no significant effect of TVNS as a standalone predictor of the two-back task performance. And then I did the same thing, interaction between the stimulation group and functional activity. There were weak significant results. But then again, I removed that outlier that was identified before, and then it was no longer even weakly significant. And again, it was close to some of those thresholds, but I don't think quite enough. Okay, so now getting on to like actually just kind of interpreting this. And this is kind of where we're going to talk a little bit about what some of those decreases in functional connectivity might mean. So as I stated at the beginning, lower functional connectivity is typically thought to be associated with lower task performance and lower, I guess, like working memory, attention, and visuospatial processing performance. However, we're looking specifically at in-task improvements or differences, really, I should say. So if we were looking at resting state connectivity, then it would be pretty, I think, reasonable to say that decreased functional connectivity could be linked to maybe decreased task performance. But because we're looking at only what's happening within the task, I would instead say that these decreases in functional connectivity may be a indicator of how much effort the participant, or cognitive effort, really, I should say, that the participant is putting in. So several studies have actually been published that link lower functional connectivity during task performance to cognitive effort needed to sustain a task. So this is not necessarily correlating lower functional connectivity with performance now, which is what we were testing throughout these hypotheses. But instead, this is saying it's possible that these lower functional connectivities as a result of TVNS might be linked to how long they can stay focused, how long they can sustain their working memory, their attention, and their visuospatial processing throughout the course of a task. And so this would potentially indicate a higher capacity for, again, sustained working memory, attention, and visuospatial processing as a result of stimulation, but would not, as we saw, necessarily say anything about the performance. That being said, though, it is also important to note that my research during this did not necessarily explicitly test for this. So as I kind of mentioned before, resting state functional connectivity analyses would be really important to test to validate this. So
do we have the ability to do that in our data set?
We totally do. Yes, we do.
We do. Right.
Yes, yeah. So that is definitely a future direction of this as well, and something I'm hoping to do next month to see if that holds up. Where if we see either no or increased changes of functional connectivity in the resting state, and then we can compare that to the decreased functional connectivity in the like task state, then we can say, heck yeah, TVNS is making it so that the participants don't have to try as hard to sustain a level of cognitive performance. So I'm very much looking forward to that. But kind of just briefly discussing some of these limitations. Again, talked about it a little bit at the beginning, but with 23 participants, these results cannot be generalized to a population, and are pretty prone to outliers as well, which I did my best to scan for, as I kind of talked about. And then another thing that I kind of just noticed is that 83% of the participants that were included in the study identified as white, and almost 70% had at least 16 years of education. So in terms of just
meaning you assessed old people in a college town?
Yep, exactly, yeah. So yeah, again, as far as generalization of these results go, may not quite be there. And then as a final limitation that I kind of just want to briefly talk about is just, again, that aspect of only having those two occasions that are separated by 2 weeks. So we're not fully able to get kind of the longevity of the effect of stimulation, or whether there are like maybe delays in effect that might happen outside of that 2 week intervention. Whoops. What did I just do? Okay, there we go. And then future work, we have some data coming in from the, or that's from the University of Arizona, that'll bring our total sample size up to 38, I believe. And then we're collecting 2 more on our end to total 40.
Indeed.
So yeah, that'll definitely be good to get in these analyses. And then again, this is just a note on testing whether TVNS is sustaining working memory and those cognitive domains of interest, as opposed to testing for task performance like I did in this.
Yeah.
So
So
yeah.
the only thing I would say relative to your conclusions is we didn't assess really. So these tasks are designed. There are behavioral differences in the tasks, but in the scanner, what they're designed to do is perturb specific networks. So as you found, there were correlations between right parietal volume, for example, and the visually spatial heavy useful field of view test, which makes sense, right? But there are better ways to test performance and get into tasks that have better psychometric qualities in this. And we do have a more extensive cognitive battery that's outside of the scanner that you can look at associations between both, of course, your vagus nerve stimulation, but also the MRI metrics to those performance changes in terms of the conclusion here. So what I see anyway from what you've presented is there's an effect of TVNS on functional connectivity, at least preliminarily in this tiny sample, right? But we don't have a great feel for the interpretive direction of that in terms of was this a good effect or a bad effect. There's some mixed findings in that regard, or at least mixed clarity. Yeah.
Yes. Yeah, absolutely, absolutely. So yeah, I think that's a great way to frame it, and I might kind of go back in my conclusion, maybe reword a little bit to kind of make sure that that is clear in that end paragraph. But other than that, that's everything that I have to report here. That is the end of my slide deck. And so I will ask whether there are any, I guess, questions. I realize we're a little bit over time, but
well,
yeah.
In fairness, we asked questions while you're talking.
That's true. Yes.
We didn't have to go through the question and answer session. I have one question though. Is you threw out a subject. I wasn't clear on how you defined an outlier, but I guess my question here is that subject that you threw out, what was their demographic? Were they an outlier in the demographic itself, or were they just a white male? How did you look into that? You know, and
I did not. I did not.
Yeah. I think, I'm sure you're fine, but as you go on and do your PhD and do amazing things, right? As you throw out these outliers, definitely be careful. And I'm sure, not saying what you did was wrong.
Right.
You do
Okay.
it all the time. It's totally fine. I just stress to my students sometimes that there's just variability that's just not captured, and we talked about this also in my class, right? About subpopulations and how do we define a subpopulation or stratification of a population, right? And so, that may be just a function of a specific demographic or a variable that you're just not capturing. yet,
Right.
and you don't have enough N for it to randomly populate, right?
I see. Yeah.
And so it's just something to think about, and just, you know, that's all.
Yeah,
Yeah.
I agree. I think that with these small sample sizes, outlier doesn't mean not real.
Right.
And it might be appropriate to remove them from your analysis as an outlier here, because maybe it suggests that if you had more data, there would be a nonlinear relationship of some sort, or you would have clusters of people that are different in
Mhm.
some way that we just can't quantify here. Another question I have for you is about parameters.
Mm.
Um, so what you presented here, I think it was, what was it, 200 microsecond pulse width, and
Ah,
2 times threshold, right?
yes. Yeah.
Right. So and then your sham approach. Can you explain the sham?
Sh- Uh, sorry for explaining the sham. Do you just mean like the study design or?
How did you do? So this was a sham control study, right?
Yes. Yes, that's correct.
Okay, and you said there's 2 electrodes that you presented, right? So you had the
Yes.
tragus
Hmm.
and then in front of it. Okay.
Yes.
Uh, so I see no sham electrode. Um, so how did you do sham?
Oh, I see. Okay, yes. Because this was a double-blinded study, we did not have access to whether the participant we were seeing in any given day was active or sham. So the electrode placements were exactly as you see here every time. So instead, what we did to provide the right either stimulation or not stimulation is we used a blinding box that was connected to the output of the waveform generator that provided the small AC signal. And so those blinding codes came from like an external source. I believe it was Damon, right, who actually generated those
Damon
codes.
built Damon built a blinding box.
Oh, he built the box.
And
Okay. Yeah.
yeah, he's a real engineer and stuff. It's
Right.
cool.
Yeah.
Right. So he built the box. There's a dummy load applied, and he's got, you enter your code so that the person doing it doesn't know if it's vagus nerve stimulation or not. Um, but what is the subject's experience? This was between subjects. So the
Right.
people, for example, in the sham stimulation group, what's their experience of stimulation?
Yeah. So they actually do not get anything, at least as far as I'm aware. And that is maybe actually another probably limitation of the study that I probably could have mentioned, is that oftentimes we will get participants who say something like, "Hey, I'm not feeling anything," and we'll have to be like, "Hey, this is a double-blind study. I'm sorry, but you can't let us know that you're not feeling anything." Um, and so, especially given that we double check the electrodes, make sure everything's running and in place, it's
Mhm.
pretty safe to say.
So, so did you have any um, questionnaire of whether the sham worked?
Um, what do you mean by work? Oh, like whether they didn't feel anything?
Yeah, or
Yeah.
well, no. So did the group that got sham think they were being stimulated?
Oh, I don't think we performed a participant side
We did.
questionnaire. Oh, we did.
We
Okay.
did.
I guess I did not maybe have access to that. So I guess that's not something that I included in this study. But I guess that would be really interesting to know um whether they thought they got stimulation affected anything. Um
Hmm.
yeah, I did not know that we collected that actually, which is a little embarrassing. So yeah, maybe I'll try to take a look at that next month as well. Run some tests on that.
Yeah, I have two questions.
Yeah.
So with regards to the stim, my battery's dying on my, how much do we got? 6%. Make it quick. Um, so
Zoom is a hungry beast. Yeah.
so with the stim, it sounds like it's a current that you're eliciting, or voltage, or what do you?
Yeah, yeah, it's a small AC current.
Okay, so you're eliciting this current, and um, do they feel that current
So
even with
this,
stim?
yes, the stimulation group does feel
Gotcha.
the yeah. What
And
we tell them, oh sorry, go ahead, go ahead.
yeah, so okay, so with regards to that, so the idea of the stim is supposed to release neuroepinephrine. So someone that would be anxious of getting shocked, right? How, and I think maybe that goes to Dr. Williamson's kind of question is if they're anxious, then they would release maybe neuroepinephrine,
Mm,
and so that may conflate this impact of the questionnaire. If they feel like they're getting shocked in the
mm,
sham, I would assume that they're releasing these catecholamines. Um,
mm.
you may still get the same effect.
I see. Yeah.
Yeah.
Yeah, that
So
would be
it's
really
something
interesting to
to kind of
test.
think about.
Mhm.
Um, I it's kind of interesting. Yeah. I never thought about, doing a questionnaire like that, but given what it's
Yeah.
eliciting, yeah.
I, I think that sham issue, and then what are we doing with the nerve, are big. I think it's too simple of a story, though it's one that a lot of people, including DARPA and NIH, have glommed onto. This isn't a recrimination of Xavier at all. To look at this as a locus coeruleus booster only.
Mm.
If that were the case, then we're going through a lot of trouble when we could just give people methylphenidate or Adderall and call it a day, right? Uh, the nucleus of the solitary tract projects into cholinergic, serotonergic, GABAergic, and noradrenergic pathways.
Mm.
And when you think about the vagus, it innervates the entire viscera. So this is the heart, lungs, GI system, all that, right? And it's an 80% sensory nerve. So we were interested, of course, in that sensory nerve pathway into the brain through all these neurotransmitter systems. But activation of the parasympathetic nervous system, for which the vagus is the primary cranial nerve, Xavier, does what?
Uh, that's like the rest and digest system,
That's
right?
right.
Yeah. Mhm.
So it's relaxation. So you have increases in high frequency heart rate variability, right?
Yeah.
Which is your heart rate slows down, the variation is more, you're resting, you're chill.
Mhm.
Uh, when you have sympathetic nervous system activation, that's adrenergic, right? So your accelerated respiration, accelerated breathing,
Yeah.
sweat response, you're hyped up, you're ready to fight or flight, right? Um, so if you only looked at that stimulation, that vagus path as noradrenergic, that's kind of confusing.
Right.
It
See.
doesn't make sense. I think that is curious, especially in the context of this. So what is vagus nerve stimulation FDA approved to do? The surgically implanted type.
Mhm.
It's epilepsy treatments, right?
Epilepsy. So it's reducing the frequency of epilepsy,
Mhm.
right? Would giving people methylphenidate increase or decrease the likelihood of a seizure?
Probably increase.
Right. And the anti-epileptic drugs often affect GABA, right?
Mhm.
Which is an inhibitory neurotransmitter. Now, they don't know why vagus nerve stimulation works to reduce seizures, but it does.
Okay.
And that's a cuff electrode, right?
Yeah.
So we have a very complex neurophysiological circuit here. And then the other thing, too, of course, is what are you doing when you stimulate the nerve? Um, are you changing the likelihood of an action potential? Are you eliciting an action potential? Are
Mm.
you interfering with the function of the nerve? That's what DBS does, right?
Right.
So you stick the electrode into the subthalamic nucleus or the globus pallidus in patients with Parkinson's disease. It interferes with the circuit, their tremor goes away, right? That's pure interference with the function of the system. Is that what we're doing? And I think neurophysiologically, we actually don't know relative to these stimulation methods how we're interacting with the circuit. People have spent a lot of money and time trying to figure that out. A whole bunch of people throwing their hands up in the air like this, but look, we get these interesting functional changes and behavioral changes. We stop seizures and cluster headaches, and could it be distraction? Or anxiety, because of course emotion changes neurophysiological state, right? So we're just prompting people to have a response. Shock, I think, is an interesting term to use. It is quite literally, right? We're doing electrical stimulation in these patients, but they perceive it as shock. So we're titrating in a way where they perceive it as comfortable, right? That's the language. So it's more like a massaging situation, and we use other electrical technologies to do muscle treatments and so on. They're not shocks per se. Though sometimes people report that they are itchy or create throbbing sensations. They're present. So that sham issue is important because we don't know how the presence of that physical feeling, which is a different nerve, right? So we're stimulating a different nerve. That's what's generating that feeling of an electrical sensation, whatever that manifest has, is a different nerve. So what is, and that's not an action potential either, per se, at least not specifically. So another question I have for you, Xavier, is about the use or deployment of vagus nerve stimulation. So in your read of the literature, in learning paradigms, let's say in rat models, and maybe this is an unfair question, I don't know, but how are they using vagus nerve stimulation?
Yeah, I will say I didn't quite see too many on rat models. Most of the literature that I looked at for vagus nerve stimulation was treating things like epilepsy in
Okay.
humans. Yeah.
So check out, if you get a moment, Christina McIntyre's work
Okay.
in rats. So in extinction learning, and then there's a whole bunch of groups that have done this where they're using it as as you describe, a norepinephrine burst. So you stimulate the vagus when the rat does something you want to reinforce the change in neuroplasticity and that learning pathway with the norepinephrine burst. So it's a paired stimulation, which is not what we did here, even though we call it paired stimulation or in cognitive training. This is continuous. So there's other questions, right, about duration of stimulation, when to stimulate, that are beyond the scope of what you've done or what we've done here to address. Yes?
Yeah, absolutely.
All right. So do you have any other questions?
I wanted to ask about the frequency and duration, and I just, why did you choose the parameters you chose? I don't think you talked about what the parameters you actually chose, but
Yeah. So the parameters, I did not actually choose them for this study. But I can tell you that the frequency it operates at, if my memory is correct, I believe it's 25 hertz. And yeah, I know that there is a paper explaining that, but off the top of my head, I cannot explain to you why 25 hertz is the frequency that we use.
And then what's the duration? How long is the pulse of 25 hertz?
200 microseconds. Yeah.
That's correct.
200 microseconds.
Yes.
Is that perceivable to the patient for?
It is, yes. Yeah.
Okay.
And it kind of feels like something like almost like tap dancing on your ear. It's a interesting sensation for sure.
By a very light person, maybe like by a
Yes.
little fairy.
Yeah.
Right.
Yeah.
I have a question. So how do you put in a 25 hertz signal for a duration of 200 microseconds? So I
The
feel like you wouldn't even get through a full oscillation. So
mhm.
the duration is 200 microseconds to,
Yeah.
yeah.
Yeah. Well, the cycle time is like, I think 40 milliseconds. So yeah, it gets through a fraction of the cycle.
Yeah.
Yeah, I mean the function or the waveform generator is pretty big. So I imagine most of what it's doing in there is just actually trying to get that out. But in terms of how that actually stays efficient with a pulse with that small and a frequency that also low. Um, yeah, I'm not
So then,
entirely, yeah.
so then every time you hit that, uh, that it stimulates you, is the phase of the signal the same, or just within the 200 microsecond pulse, you're at different phases of the 25
I
hertz?
believe the phase is the same. Let me I actually might be able to check that super quick. I may have
That
written
would
that.
introduce a ton of additional variability.
Let's see.
Yeah. These are just things to, I'm kind
Mhm.
of slightly curious. I don't want to keep you guys,
Right.
but just
Yeah.
things to think about.
Right, for sure, for sure.
The dosing space is a big issue. So there's been a few dosing studies. We did one recently that's about to be submitted in sleep across 9 different parameter combinations, alternating frequency, pulse width, and level setting. Somebody else did a similar one at heart rate changes. And in general, the wider pulse widths cause more of a reaction. I've seen them up to 500 microseconds in this space, but they're also far less comfortable. So you can't have so there's an interaction between the level setting and the pulse width and the frequency where people will not tolerate the wider pulse widths. So in our dosing study, the dosing that actually survived the regression model against sham was 100 microsecond pulse width with a 20 hertz frequency set to 80% of the threshold of discomfort, which is a completely different level setting approach here. We had picked when we designed this study the 200 microsecond pulse width before we did that dosing study, and that was based off of another study that showed these wider pulse widths worked. And we had done a prior one where people had shown functional connect or bold response differences in limbic circuitry, which is important in memory consolidation in using 100 microsecond pulse width and a 20 hertz frequency. We published one in MCI with a 50 microsecond pulse width and a 10 hertz frequency. This was in during fMRI scanner and or during fMRI stimulation, and the stimulator quality for what was MRI safe was not capable of doing the higher frequency and pulse width combinations. So we throttled it down based off of the lit in terms of what people have published showing any sort of physiological signal. But I would consider the dosing and duration issues to be critical flaws in the current literature with respect to understanding physiological responses. The one, like even nerves habituate. Your experience of the stimulation from a sensory perspective habituates. And there are data with other types of stimulation types that you need a more stochastic stimulation pattern. And one of our colleagues, engineering colleague, Matt Schieffer, has been recording firing of the vagus at the gut and then modeling his stimulation paradigms based off of the firing of the vagus, the pattern of the firing of the vagus in specific physiological states. So he's going after eating behavior. So if you've got a time when satiety has been reached, what is the vagal firing at that time? If you introduce that firing earlier in the eating behavior, does it stop it? This sort of question. Yeah.
Yeah, cool stuff. Cool stuff.
Cool.
All right.
All right, so let's have Xavier step out here.
Yep.
So give me a host again, and we'll throw you in the waiting room.
Sounds good. I'm gonna go ahead and